Good morning, ladies and gentlemen. My name is Maysoune Charef. I'm a PhD student in applied microbiology. I would like to share with you a little part of my work entitled Preliminary Profiling of Pycnolytic Bacteria from Potato Tubers. Our presentation will be in accordance with the following plan. We start with the introduction, where we will talk about the objectives of our research. Then we're going to represent the different methods and materials used to carry out the work. After that, we're going to talk about the results obtained, and we're going to end with the conclusion. As we all know, potato is a traditional widespread crop. It is the third crop in order of importance after wheat and rice. Unfortunately, this tuber is the target of many bacterial diseases, and this may threaten the food security of any country or region. It is therefore absolutely necessary to highlight the associated microbial communities to take into account the diseases affecting its production. Here we're going to take a look at the potato morphology. This picture is showing a longitudinal section of a potato tuber. As we can see, there are apical bud, lateral bud, prederm, lenticels, and the stem end. Among potato diseases, there are soft throat and plague leg, due to the genus of Pictobacterium and Dickia. There are gram-negative, non-sporulated mobile bacteria. They are characterized by fermentative metabolism of respiratory type, aeroanaerobic optional, and they have a strong pycnolytic activity due to the production of pectins. Our objective was to deepen our knowledge of the biodiversity of microorganisms present in the potato register. For the microbiological identification, isolates were isolated from potato tubers diseased, showing symptoms of soft throat collected from the markets. The sampling was done according to the following protocol, and for the identification of the bacterial strains was based on the determination of morphological and biochemical identification. For the microscopic examination, study was carried out taking into account the form, the contour, the relief, the size, the surface color, and the porosity of colonies, and the microscopic examination was carried out according to the gram strain. And this picture is showing the diagram of the gram strain. For the biochemical identification, we done three tests: catalase test, oxidase test, and nitrate reduction tests. And all the protocols are shown in the following diapositive. And the results are shown in the following pictures. For the results, as we can see, this graph shows that the number of gram-negative bacteria is higher than the number of gram-positive bacteria. And this graph is showing that most bacteria are oxidase negative and catalase nitrate reduction positive. As a conclusion, we can say that the isolation carried out on a total batch of 13 tubers sampled on the basis of microbiological and biochemical identification generated on the tubers, we were able to identify that most bacteria are gram-negative, oxidase negative, catalase positive, and nitrate reduction positive. These results confirm the presence and diversity of bacteria in potato register. As a perspective, we would like the data collected during the study as well as the conclusions drawn would constitute a basis of study for future work in order to better explore the different aspects related to potato community. And thank you for your attention.